right, well, thank you everyone for joining today. For those of you who don't know me yet, I'm Alex Lemens, the director of our Molecular and Genomics Informatics core. We also have online today Pat Soteropoulos from the Genomics Center, who most of you probably met at this point. She has the full suite of Illumina sequencers, everything from the MiSeq, MiniSeq, NextSeq, NovaSeq, and regularly handles all of the single cell workflows at Rutgers NJMS, which is great because today we have the Miltenyi team who's going to be talking about their sample preps to improve single cell quality, which is something that has been so critical in so many experiments and something we really need to kind of optimize for a lot of projects going forward. But before I turn it over to their team, just the general housekeeping items that we kind of go over every time. If you have any questions, please drop them into the chat or the Q&A box. I will monitor for it kind of on the fly if we can. Anything that's not mentioned during the talk, we will ask at the end. So with that, I'll turn it over to Snyder and the Miltenyi team to talk more about their sample prep for improving single cell quality.
Hi, thanks, Alex. Hello, everyone. I really appreciate you taking the time out of your day to attend this seminar. I hope that everybody's having a wonderful week so far, and hope that there's lots of excitement for the start of this new academic year. As mentioned, my name is Snyder Desir. I am the new local Miltenyi rep for Rutgers University, Newark in particular. And today, I'm here to speak with you about our Genomics tissue dissociation system and how it can help improve your sample preparation for single cell sequencing. Now, if you're not too familiar with Miltenyi, just a quick overview to get us started here. The company was founded in 1989 by Stefan Miltenyi, and today is Germany's largest privately owned biotech company operating now on a global scale. We have over 3,000 employees, and we're in more than 50 different countries. We do place a large emphasis on research and development, not solely for the manufacturing of lab products, but for the development of therapies. As most people don't know this, but we are in fact actually also a clinical company, and we have over 65,000 cell therapy procedures being done in the clinic using Miltenyi's product every year now. And so what started really as just a cell separation technology startup have now blossomed into this large translational biomedical research and cell therapy organization with more than 30 years now. And in the space, we've done a lot in the sample prep and cell separation space. And I think this places Miltenyi in a fantastic position to address some of the sample prep demands of doing single cell RNA-seq. As most of you know, single cell genomics is very popular nowadays, and for great reasons. It's a very popular technique because we have the ability to study gene expression dynamics on a cell-by-cell basis and really get a great picture of cellular diversity in the tissue, as opposed to, say, doing mean expression from bulk and sample analysis. But in order to take advantage of the transformative power of this technique, it does demand that you work with properly prepared single-cell solutions. Now at Miltenyi, we've specialized in delivering quality samples for basically any immunology applications. And this becomes even more critical, I think, when working upstream of a more sensitive endpoint like genomic outputs. A lot of time and a lot of money are put into sequencing experiments. And so having high quality samples to work with, I think, is really key in saving both. For starters, good quality samples needs to be debris-free, as cell fragments and sticky material like free DNA can cause aggregates and really throw off your cell counts. You also want to reduce dead cells as much as possible, especially because dead cells can easily lyse and release ambient RNA that's going to contribute a lot more to your background and really compromise the quality of your data. Overall, what you really want are cells that are simply happy, stress-free, with undisturbed markers and nuclei. It's also very important to have preserved native gene expression profiles that wasn't significantly influenced by a harsh dissociation method. And so one of the comprehensive portfolios that Miltenyi provides is a simple prep workflow that addresses these requirements right from the moment you harvest your tissue. This, starting on the left here, begins with the max tissue storage solution, and then going on to having an automated tissue dissociation system, and then materials for cleaning up that sample, as well as other instruments and reagent that you'll want as part of your process to ensure optimal sample quality and concentration before going into your library preparation. So if you're not processing your tissue right away, we have a tissue storage and solution that's comparable to human transplant organ reagents. I know sometimes samples are harvested at a different location or needs to be transported to, say, a core facility for processing. And for these situations, the tissue storage solution is perfect because it's going to preserve both viability and compositions in your tissue. This is one of our validation studies where we looked at these parameters and dissociated mouse tumors via flow. And what we found, starting with the graph on the left here, was that after 24 and 48 hours, there is no substantial difference in the viability of cells from samples that were stored with our max solution compared to cells from tissues that were harvested fresh. When looking at the immune cell composition in the middle graph, you can see again, even after 48 hours in storage with our solution, there's no loss in these tumor-associated immune cells. And so we're still able here to preserve your CD4s, your cytotoxic T cells, CD19, and others as if the tissue was processed fresh. We also looked at cellular stress using a QIAGEN stress gene profiler panel. And again, we had no significant upregulation of these stress-related genes in the store samples compared to fresh samples over the 2 time period. And if you're wondering about tissue type, this reagent has been validated for several different tissues. So whether you're working with animal samples, human biopsies, or any kind of, say, resected tumors, your samples will be kept as good as fresh for up to 48 hours with our max solution. And going over this one real quick, I want to mention, in addition to the 4-degree tissue storage solution, we also provide solution for freezing cells in solid tissues that you'll need to preserve for over a longer period of times. So now we can get into the core of the sample prep workflow, which is efficiently extracting cells from tissue for your sequencing application. And the best technology in the market to perform this task is our Genomics tissue dissociator octaweight heater system. With this system, you'll have the ability to generate single cell suspension with a very little effort in a way that's almost as simple as running your household blender. This instrument solves a couple of things. Manually disrupting tissue and mixing in your own cocktail of digestive enzymes can be extremely harsh on cells. Not only that, the process of doing this manually can often be labor intensive, requires a certain level of expertise that's tissue specific often, and the results can vary between users. Some of you who are users and may already be familiar with the capabilities of this system knows that the Genomics enables you to fully automate and standardize tissue processing. With this, you can dissociate and or homogenize 8 different samples at the same time, each running on their own independent program with or without individual temperature control for a particular protocol being run. This instrument also comes preset with protocols that let you process several different tissues. And if you're working with some rare sample for whatever reason, we do give you the ability here to create your own protocol and save it on the instrument. For dissociating your tissue, the Genomics basically combines the mechanical and enzymatic tissue treatment process and lets you, in a walkaway fashion, obtain your single cell suspension. The system allows researchers at virtually any level of expertise to process tissues automatically in a very fast, gentle, and reproducible way. In fact, if you were to compare the recovery of isolating cells using the manual method versus our Genomics system, it's actually pretty night and day. This is data from a customer who compared to a Treg isolation that they've done manually to that of using our Genomics system. And what this does is here really give you an example of how efficient the system is in pulling cells from the tissue. They were able to get this amazing increase in their Treg yield here after switching to using our system and applying the tissue-specific protocol. And one of the reasons behind this high efficiency really has to do with the way the tubes that fits into the Genomics system are designed. There are two tubes that comes with the Genomics system, and they're based on the downstream application of interest. There's the molecular application tubes, or the M tubes for short, which are used to homogenize tissues and enables you to obtain samples for isolating things like mitochondria, RNA, proteins, and other subcellular contents and organelles of interest. And then there's the cellular application tubes, or the C tubes, that are used to gently dislodge cells from your tissue and leave you with a well-dissociated single-cell solution. To take a look at how this is done in a little bit more detail, we can zoom into the C tube cap and really get an idea of how it's engineered to do this. Now, inside the cap, where you place your tissue, you'll find that there's these rotors and stators, with the rotors being the part that's actually mobile. What's done here is that there are these tiny spacers that's incorporated into the stator component and a well-defined distance between the stator and the rotor, so that as the rotor moves, instead of grinding the tissue harshly against the stator, there's simply a gentle shearing force that opens up the tissue, which allows the digestive enzyme efficient access to the tissue matrix in a way that lets you basically dislodge the cells out of the tissue gently. And so your cells here aren't physically being damaged by a mechanical process, and you do not have to use a boatload of enzymes to digest them out, so you avoid major off-target tryptic activity and preserve important epitopes and markers. So happy cells. Next to the instruments and tubes, we have the tissue dissociation kits. And aside from their efficiency and consistency, these tubes were validated while testing their enzyme mixtures against over 200 important markers. And what this means for you as a user is that you'll know if and how your epitopes are being affected by our kits. And so if you're interested in, say, indexing transcriptomes and epitopes to capture RNA and surface protein expression of a single cell, you can take a look at our application note on a particular kit. And if you need to tweak your protocol for that application in any way, you can contact us and we'll get you expert level help to troubleshoot your experiment. Now, what I'm showing you here are just some examples of our kits. We can virtually help you process any type of tissue. This is part of an entire library of kits that we have. And if you go on our website, you can find out more, or you could just reach out to me if you need to talk about some tissue type that you're not seeing here. Okay, so what I've talked about today so far is that basically the combination of the 3 Genomics system, which are the instruments, the tube, and the tissue-specific kits, when put together, will dissociate your tissue gently with minimum stress on those cells. Will preserve your epitopes and guarantee that this is the most efficient tissue dissociation process. And again, because this can all be done in a fully automated walkaway fashion, you'll reduce your hands-on time and decrease user variability. What I have next is just a quick video of this system and at work. hopefully this plays here. All right, so this is just a quick video. It's showing you some of the materials here, and there's not really anything extra that you need, just items that you have every day in the lab. Quick summary of what's happening. Basically, you first reconstitute the enzymes that comes with the tissue dissociation kit, and any unused reagents here can be stored up to 6 months at -20. Basically, after you reconstitute the enzymes, you enmix your tissue, as you see here. And once that's done, you add a certain amount of media to the tubes. It is a protocol. After you do that, you transfer in your cocktail of enzymes. And from there, you would add in your tissue samples. You cap the tube, flip it over, and make sure the tissue settles to the cap. You place that in the instrument, you pop in your heating sleeves, and you run the tissue-specific protocol. And that's pretty much it. You can, after this, go run, have a cup of coffee, go to lunch, go to a meeting until you have to come back to do your sample cleaning, which is what we'll get into in the next couple of slides. Oops. Okay, so now that we've generated our cell solution, we need to clean up and get rid of any unwanted things like debris, cell aggregates, and other materials that can really throw off your sequencing and probably clog the microfluidics in the instruments. And for that, we have here, starting with our strainers and filters, the ability to remove larger materials out of those samples. These filters and strainers comes in various sizes, ranging from 100 down to 20 microns in pores. These will fit perfectly into your conical tubes, and they can stack on top of one another for one-shot filtering, and they won't clog because of how well they're ventilated. I'll just run this other video here to show you how they perform against the standard cell strainers that you're normally using in a lab. And if you go buy these strainers from us, they cost just about the same as your regular cell strainers, except obviously, as you can see here, they work much, much better. And if you need to, you can just reach out to me for a quote. We also have a range of removal reagents, and these include solutions for removing smaller debris, red blood cells, and the removal of dead and dying cells. Other than that, you can also find, when applicable, reagents for the removal of myelin and endotoxins from your sample. And having these reagents as part of your sample prep workflow is really critical because filters won't be effective in removing some of these smaller contents. And it goes without saying, especially that you want to remove dead and dying cells, as well as red blood cells from your samples, because these can significantly impact the quality of your sequencing data later on. So in this study that we did in collaboration with 10x Genomics, we looked at how the addition of some of these reagents can improve your data quality. What's done here was that cancer cells were injected to generate models of breast, colon, and melanoma tumors in genetically identical mice. This was done using 3 animals per tumors, and then the tissues were resected from each group for a total of 9 tumor samples. Subsequently, the tumors were dissociated, of course, using the Genomics octodissociator with heater system and the appropriate tumor dissociation kit. And then the samples were taken through different cleanup processes using our removal reagent prior to using the Chromium single cell prime 3 V2 library kit. And what we ended up doing here was making 47 sequencing library and sequencing over 84,000 single cells. Condition A here is just the cleanup step with the filters. So this was using a 70 micron smart strainer. And condition B is adding the red blood cell lysis step. Condition C adds the dead cell removal step. And because tumors have a lot of dead and dying cells, whereas in some cases, we actually find that a single round of dead cell removal kit was not enough to yield the desired viability, we added a second round of dead cell removal that we're calling condition D here. So when you run the 10x Cell Ranger software, there are a couple of metrics that are reported back to you so that it can help you determine the quality of your starting material. One of these metrics is around library cleanliness that's shown here on the left. And in these analyses, all of the different replicates were combined together. And what we're looking at here, when looking at on the y-axis, we're seeing the fraction of reads that can be confidently mapped back to a single cell versus the background or just say free floating mRNA. And on the x-axis, we have the different cleanup conditions that were applied. You can see that condition A, when using only filters, about 80% of the reads were cells, which mean 20% of these reads are just being thrown away. By adding the red blood cell lysis and the dead cell removal steps, we increased the fraction of reads to over 90% here. Next, over on the right graph, we evaluated library complexity. And this was done by basically calculating the median number of genes detected per cell. And on the y-axis there, because the number of genes detected per cell is affected by sequence and depth, we normalize each library to 20,000 reads per cell. And once again, you can see from the graph that there's an increase in the number of genes being detected here with the addition of each cleanup steps. So we've so far covered tissue dissociation and cleaning up your samples. Now we can get into isolating specific cell types of interest. Now, often prior to library preparation, you may want to isolate and enrich a cell type to maximize the number of a particular population you want to sequence. For this, cell sorting is usually the popular method, but another easier and even more cost-effective option is to use our Max microbead technology to isolate your cell of interest. This is basically the same method used to do dead cell removal, except the difference here being that the target cell of interest remain untouched. So what that means, for example, is that if you wanted to, say, sequence neurons after doing a brain dissociation, you can use our Max technology to enrich them and avoid background data that's going to come from your non-neuronal cells. And what we'll do in that process is basically magnetically tag those unwanted cells and have them remain in a column while your flow through becomes the fraction that's highly purified with your untouched neurons. And this was done in another study in collaboration with 10x Genomics, where we added this step of enriching neurons after doing a brain dissociation. The workflow here, of course, starts with the Genomic system, and we use our adult mouse brain dissociation kit to do this. The cell suspension was subsequently filtered and cleaned with RBCs and dead cell removal kits, and then neurons were isolated using our neuron isolation kit. First, showing you in this UMAP clustering plot results from the cell suspension before neuron isolation kit was used. If you focus just on the red circle here, you can see that there's a very low population of neurons, and we're only getting about 5% of these cells here. And this was confirmed with flow data over on the right. As expected, when looking at the clustering, other cell types being identified here, so astrocytes, oligodendrocytes, microglia, and things like vascular cells, really overwhelmed and masked the data from our cell of interest, and we can only make the distinction between mature and immature neurons. After using the neuron cell isolation kit, our cell of interest here was enriched to over 90%. And what's fascinating is that without the other cells masking the data, we're able to analyze significantly more neurons in this library. So we get an extremely clear picture here of the heterogeneity of the neuronal cell population. And if you take a look at the two data side by side, you can see how we went from just seeing mature and immature neurons to now identifying and distinguishing between 11 neuronal cell subpopulation, which shows just how effective MAX technology can be at improving your library preparations. In addition to the neuron isolation kit, we also have microbeads for almost every cell type you'd want to isolate for your starting sample. So please, if you have any questions, email me, go to our website, and we'll be more than happy to help you. Now, with regards to separators used with our MAX technology, we offer a wide variety of options depending on your throughput or needs for automation. Starting with on the left here, we have our manual separators like the MIDI and QuadraMax that can hold a small number of samples. And in the middle is our AutoMax Pro that's fully automated and made to run up to 6 samples. And then there's the Multimax 24 and the Multimax X, both of which processes up to 24 samples. And the only difference between those two is that the Multimax X is fully automated and is basically like the Multimax Separator Plus here, but it's surrounded by robotics to do the work. And all of these options can be used to isolate any of your cell type of interest. However, for some application, you may want to opt for using a cell sorter to isolate your cell type. And droplet sorters are very common, but these sorters charge and decompress your cells and apply a lot of pressure that can be really stressful on your sample. An alternative way to do this is to use a microfluidic sorter like our MaxQuant Tito. Due to our microfluidic system, there's no charge and decompression being applied here. And so this results in a more gentle sorting process for your cells. So at the heart of this instrument is our Tito cartridge that's going to do the job of separating out your cells. At the bottom of this cartridge, what we have is a microvalve controlled by a smart chip. And as the cells are being interrogated by the instrument's lasers, the chip will recognize the fluorescently labeled cell of interest and triggers the valve to open and direct that cell of interest towards the positive collection chamber in the middle. And because this sorting is being done solely within the cartridge, which comes wrapped, and your sample is being prepared and added to the cartridge in the hood, this is fully sterile and requires very little technical expertise, and there's no carryover or contamination. Now, I'm not going to go into too much detail about this instrument. We have a whole family of MaxQuant technology that could be talked about, but that's well beyond my expertise. We do, if you are interested, have another seminar coming up on September 7th that I'd like to just pitch real quick. This is going to be at the Cancer Institute of New Jersey, and this is going to be done by my colleague Natasha, who's like a 7-degree black belt in flow cytometry. And so if you want, you can reach out to me or Natasha for information on how to register for that. And with that, what I'll do here is just kind of wrap up with a quick summary. One being that it's very critical to prepare high viability cell solution that's debris-free prior to your library preparation. So our Genomics technology will help you do that. You'll combine the mechanical and enzymatic tissue dissociation process and obtain your single cell solution in a very easy walk-away fashion. We have removal kits that's very effective for increasing your sample quality. And finally, we have various ways of isolating target cell types of interest with our MAX technology to not only increase the sensitivity for your analysis, but also do this at a decreased cost. So finally, I just want to say thank you to everyone that's here and go on to address any questions that you may have.
Yeah, thanks for a great talk, Snyder. Yeah, if anyone has any questions, feel free to drop them in the chat, the Q&A. I know I've got a couple real quick. So I guess first, with a lot of the kind of what I call single cell plus methods, all these extensions into site-seq and hashtags that require antibody tags when you're doing the library preps. Is the MAX workflow compatible with those types of other antibodies?
Yeah, so I wish my manager Jim was here to provide a good answer for that. I'm not too familiar with that, and so I don't want to say anything that's gonna get me in trouble. I don't know if any of my other colleagues are on the call and can kind of pop in and jump in for that.
Yeah. Hey, hi, Snyder. It's Michaela Hernandez.
Oh, hey, I didn't know you were.
Hey, everybody. Thanks so much and great presentation, Snyder. So thanks for that question. So, as I understand, so your question is if you're using the MAX technology, like the separation beads or things of that nature, is that compatible with any other like need for, let's call it additional labeling downstream, right? Mm-hmm. So the answer is yes, absolutely. So the Miltenyi bead technology is such that we really very minimally label the cells of interest. So that's why, as Snyder was talking about our separations technology, why we use the columns with the magnets that allows us to very minimally label and separate cells, leaving plenty of epitopes for downstream processing.
Okay, perfect. Thanks.
Sure. What other questions?
I've got another one if nobody else has one. So the freezing storage protocol that you mentioned kind of earlier in this, where you're freezing like the entire tissue block and then later doing the dissociation. Have you done any kind of validation assays or anything like that to determine how long those tissue blocks are sustainable for?
This is for getting for storaging cells and then doing your preparations like a couple of days later or very long term. Sorry, can you clarify that?
Yeah, I mean, I'm thinking more like months, years later, possibly. Like especially if you're doing like a patient cohort where you may be collecting a tissue block on, in August, and then maybe down in February is when you're actually going to be doing the dissociation library prep after you've collected a few more patients as well. Have you done any kind of validation to see if it's still, equivalent over that time point?
Yeah, so you're definitely not going to have anything like the quality that you'll get, say, 2 days later, right? That I've shown you earlier. With these longer period of storaging method, you're gonna definitely have a little more compromising of your tissue sample block there, right? So, although we have this freezing solution for longer storage, it's not going to give you there's no solution that's going to give you the same kind of viability and composition that you'll get when looking at storaging for just those 2 days.
So what are you guys doing? Can you tell us a little bit about what you're doing now and what you've seen?
Yeah, so I mean, right now we're evaluating a couple different options for these types of long-term storage projects, particularly things like fixation protocols. I know we're
Okay.
exploring like Parse Biosciences. 10x recently released their at least human one. I think the mouse one's still TBD on when that's being announced or released officially. But that's been kind of the most common thing. Or the other thing is, people rushing to get to the 10x machine, where they'll have a patient sample, and they try to go as fast as they can from harvesting the sample to the 10x machine and then bring it to the sequencing core.
Right.
So
Right.
yeah, that's the most common solution.
You guys are ultimately like looking at methods for different storage, both kind of near term and longer term, fundamentally. Okay.
Yeah.
Maybe Snyder, maybe we can put together maybe a little bit of the data that we have kind of together with some of the solutions that we have.
Okay.
Yeah, I can send you a slide with I'll go grab the data and send you some slide. Just email me.
Okay, that'd be great. Thanks. Then I guess kind of last question I have that's kind of paired off that. I'm assuming the answer is probably yes, but this is viable for kind of any single cell workflow downstream, whether it be Parse or 10x or BD's Rhapsody, anything like that. Correct?
Yep.
Okay, cool. Just wanted to make sure. Yeah.
What are you guys using? Can you give us any? And sorry, Snyder probably already knows the answer to this, so apologies. But
Yeah, we're primarily using 10x. We are currently evaluating Parse and looking at a few others as well, just to kind of see what's out there and see if there's any improvements we can bring to the investigators we work with. So we're always looking for what's the latest and greatest, but right now dominantly 10x.
great. Now, you guys primarily look at cells. Are you also looking at nuclei? Like what's?
Yes.
Yes, nuclei.
Yes.
Yeah. We do.
Go ahead, Alex.
Yeah, predominantly cells. We do have an interest in nuclei, but historically speaking, nuclei has been problematic. So that was something. Yeah.
Let me guess.
That is
Go ahead.
something we really need a good solution for. So, if we can help people get better nuclei, that would really change the game, right?
Okay. So when you say better, do you mean of nuclei integrity or cleaner samples? Like what's better to you?
Both.
Both, yeah.
Okay. And then so are you doing nuclei from all sorts of different cell types?
Yes, which is one of the other compounding factors. It seems like each tissue type requires its own kind of customized nuclei isolation,
right.
Especially to get it clean without ambient RNA, ambient DNA, and intact nuclei.
Okay. So we definitely have some potential solutions there. I know Snyder mentioned there's another talk that's going to happen next week, which is focused a bit more on flow sorting, but we're going to cover some sorting for downstream single cell genomics and things of that nature, including nuclei. So it might be interesting. So we've also got we've got some multiple solutions there. That we can talk about. So I don't know if everyone on this call has received the invite to that discussion.
Okay. If you could send that link to us, we can send it out. We usually send a follow-up email to everyone, so we can send that link to everyone.
We will do that.
Yeah.
Snyder, anything to add there?
No, I was gonna say that I already told Alex about this, that we also have a couple of seminars on YouTube. And there are some that actually addresses nuclei isolation. So
yeah, I think we can talk about. Yeah, I think we could add maybe a little bit of that discussion in with the discussion that we're doing next week, right?
Yeah.
And that would be a good time to that way people can that way people can ask questions. Oh, yeah. Thank you. Yeah. So Snyder will make sure to send everybody this. And then I think there's also a link to register as well. So maybe we'll make sure to send that.
Okay, perfect. Thank you.
Yeah, absolutely.
Yeah. And it doesn't seem like we have any questions coming from the crowd. Pat, do you have any questions?
No, Alex, you pretty much covered the questions that I had. These are the things we've been talking about quite extensively lately.
Mm-hmm.
Great. The great news is I think we have some pretty good options there. So hopefully everyone will be able to make it next week. If not, we can definitely be available to follow up for individual discussions.
Okay, Sounds good. Well, then I guess thank you everyone for joining today, and thank you for the great seminar. This is very informative and definitely learned a lot. So yeah, if anybody has any questions, feel free to reach out to myself, Pat, or we could give you the contact information for the Miltenyi team. And yeah, we look forward to seeing everyone at the next tech seminar series in 2 weeks for Twist. So thank you everyone. Have a great day.
All right.
Thanks,
Alex.
everybody.
Bye.